Endomembrane trafficking. Endomembrane trafficking literally means the movement of materials inside or within a membrane. The endomembrane system is composed of interrelated membranes within the cytoplasm of a eukaryotic cell. These membranes divide the cell into functional and structural compartments called organelles. This system includes the nucleus, rough endoplasmic reticulum, or rough ER, Golgi apparatus, secreting vesicles and other structures derived from them like lysosomes, and the plasma membrane. It is this membranous system that works together in the synthesis, modification, sorting, and transport of proteins. Some proteins will be used in the cell like an enzyme, or secreted out of the cell like a hormone. Endomembrane trafficking begins in the nucleus, where the transcription of DNA takes place to form messenger RNA. This is essential as DNA is too large to leave the nucleus, so in order to get its information out of the nucleus, transcription must take place to create the smaller molecule of messenger RNA. Messenger RNA leaves the nucleus through a nuclear pore into the cytoplasm. It can either adhere to a ribosome in the cytoplasm, also known as a free ribosome, or adhere to a ribosome on the rough ER, also known as a fixed ribosome. Here is an up-close view of what takes place on a ribosome. Once the messenger RNA strand reaches a ribosome, translation takes place. Translation involves transfer RNA carrying an amino acid coming along and binding its anticodons with the complementary codons on the messenger RNA strand to create a peptide bond. As this occurs over and over, a polypeptide chain, or protein, is formed. Once translation is complete and a protein is formed, it has two paths. If it was formed on a ribosome that was in the cytoplasm, it will be released into the cytosol to be used within the cell. If the protein was formed on a ribosome that was fixed to the rough ER, it will travel through the cisternae of the rough ER and undergo further modification called post-translational modification. The modified protein will pinch off, forming a transport vesicle. The transport vesicle leaves the rough ER and arrives at the cis face of the Golgi apparatus. The cis face is the side of the Golgi apparatus that is closest to the rough ER and is actually made up of several vesicles that fuse together to create this receiving side of the Golgi apparatus while releasing their contents into the cisternae. As the protein moves through the cisternae, it undergoes further modification where it is labeled for its targeted destination. As the protein reaches the other side of the Golgi, called the trans face, it gets packaged into a vesicle. One type of vesicle that pinches off is a lysosome. The lysosome stays within the cell and functions as the digestive system of the cell. Another type of vesicle that pinches off is a secreting vesicle. A secreting vesicle leaves the trans face, or shipping side, of the Golgi apparatus and binds to the plasma membrane, where it releases its contents to the outside of the cell through a process called exocytosis. And that is endomembrane trafficking. I hope you enjoyed this presentation.